Assalamualaikum and good morning, everyone. So today we'll be learning about the neuromuscular junction as well as the muscle contraction, which can be explained by the sliding filament hypothesis, and the Ratchet mechanism. So first things first, neuromuscular junction. So you know that this is an axon terminal, of a motor neuron axon. Now the word here is motor, and neuron axon. So motor neuron axon means, is the end of an axon that is attached to a motor, and this motor can be, one example is the muscle cell, right? Okay, now we have a diagram here which show a polarized neuromuscular junction, or neuromuscular junction that is at a resting potential. Now neuromuscular junction, is the synapse that is formed between the axon terminal of a motor neuron and a muscle fiber. That is neuromuscular junction. Motor end plate. So before I go to motor end plate, so apparently this is the neuromuscular junction. And then plasma membrane of a myocyte is known as sarcolemma. This name given for the plasma membrane of a muscle cell. Now motor end plate is the region of the sarcolemma, which is located under the axon terminal of the motor neuron. So apparently, if this is the sarcolemma, this would be the motor end plate. You can see here, this motor end plate again is connected, through sarcolemma to the T tubule or transverse tubule. And this transverse tubule or T tubule, it increases the surface of the sarcolemma. So we have here sarcomere, myosin actin, Z line. We go on that part later. Now this is the structure of a skeletal muscle fiber. You can see that the skeletal muscle fiber has many myofibrils. One tube of this is the myofibril. And these myofibrils, they are wrapped by the sarcolemma or the plasma membrane. And this is the nucleus. And the basic unit of a myofibril is the sarcolemma. So this is one sarcolemma. Now this is the T tubule. And this T tubule is connected with the terminal cisternae. You can see this terminal cisternae. And two terminal cisternae with one T tubule will make up a triad. Now a terminal cisternae with another terminal cisternae, they are connected with the sarcoplasmic reticulum. Now this sarcoplasmic reticulum, it regulates the calcium ion concentration in the sarcoplasm or cytosol or cytoplasm of a striated muscle cell. All right. So we have this sarcomere. Then this is how a sarcomere look like. So candidates, you must be able to distinguish this diagram here. This diagram here represent a sarcomere. And then you can see here, this is the M line, This is the Z line. Okay, Z line. And then we have many thin filaments. Okay, this not so dark. Okay, this light band. These are the thin filaments, also known as the actin. And these darker bands, okay, they are the um, thick filaments or myosin. Okay, sorry, these darker bands here, okay, they are the overlapping of the thin and thick filaments. Okay, now let's take a look into a more detail of the one sarcomere. Now, one sarcomere is the repeating unit of a striated muscle, okay, and delimited by the Z line. So meaning to say one sarcomere, okay, is from one Z line to another Z line of a muscle cell, okay. And then in the sarcomere, one sarcomere, you can see, okay, A band and I band. There are two regions here, the A band here, and the I band here, as well as the I band here. Okay, there's two regions of A band and I band. Okay, the A band will give you the dark band that you saw earlier. Okay, and then the I band is the region here will give you the light band when this muscle cell is being observed under the TEM. Okay, now you can see that one sarcomere is delimited by the Z lines. All right, so what is this Z line? Z line is the protein that holds the thin filament. Okay, so you can see this is the thin filament, or also known as the actin. Okay, now this thin filaments of actin is a globular protein that links two chains. You can see there are two chains here, okay, which twist helically, okay, about each other, forming a microfilament, or we can see there are many microfilaments here. Okay, and these microfilaments also known as actin filaments, okay, in muscle as well as other kind of cells. Then we have this thick filament, okay, which also known as the myosin. Now, myosin is a type of motor protein, okay, a type of motor protein that associates into filaments, okay, that interact with the actin filaments to cause cell contraction. Then we have this M line, okay, in the H band. This M line is a protein that holds the thick filament. Okay, it holds the thick filament. Okay, next we have this H band. Okay, this H band is the overlapping of the thick filament. Right. Next we have the A band. A band is the overlapping of the thick and thin filaments. You can see how these thick and thin filaments, they are overlapping with each other. This forms the A band. Okay, then we have the I band, which is the overlapping of the thin filament. Okay, so we can see, okay, this is a myosin molecule that has tails with heads. Okay, and this head of a myosin molecule, it has ATPase, which is an enzyme. Okay, ATPase means an enzyme that hydrolyzes ATPs into ADP and pi inorganic phosphates. Okay, so one thick filament has many tails and myosin heads. Then this is thin filament. You can see, okay, how this globular protein, they are twisted helically. Okay, and this is a TEM, okay, of this sarcomere. You can see how the thin and thick filament, they are overlapping with each other. Okay, now let's further continue with propagation of action potential at the neuromuscular junction, which will lead to muscle contraction. Okay. Okay, now arrival of impulse at the synaptic node. Okay, at the synaptic node of a motor neuron or neuromuscular junction will cause the synaptic vesicles. You can see the synaptic vesicles here. Okay, to release acetylcholine. Okay, acetylcholine. Mind the spelling acetylcholine to the synaptic cleft. Okay, now this acetylcholine will bind with the acetylcholine receptor, okay, on the sarcolemma, okay, or on the motor end plate, okay, and this will cause the sodium ion channels to activate, and this will cause influx of sodium ion into the sarcolemma, and sarcolemma is depolarized. Okay, now depolarization, okay, at the motor end plate, right? Now sarcolemma, this is, yes, a sarcolemma, but to be more specific, this is a motor end plate, which is a specific part of the sarcolemma. Okay, so now this motor end plate or this sarcolemma is depolarized, and the depolarization will propagate the action potential all the way, okay, to the T tubule, and then eventually to the sarcoplasmic reticulum. Okay, this is the sarcoplasmic reticulum. Okay, next. Okay, this will trigger the opening of calcium ion channels. Okay, the calcium ion channel will flow from the sarcoplasmic reticulum into the cytoplasm or cytosol. You can see, okay, how this the propagation of action potential to the sarcoplasmic reticulum, okay, will activate, okay, the opening of the calcium ion channels, and this will allow, okay, flowing or influx of calcium ion, okay, from the sarcoplasmic reticulum into the cytosol. Okay, now voltage gated calcium ion channel in the sarcoplasmic reticulum also activated or activates. So when these voltage gated calcium ions channels are activated, okay, or activated, calcium ions will be pumped, okay, from the sarcoplasmic reticulum into the cytoplasm. Okay, the accumulation of calcium ions in the cytosol will initiate the muscle fiber contraction. So again, like what I mentioned earlier, cytoplasmic reticulum regulates the calcium ion concentration in the cytoplasm of striated muscle cell. Okay, so now we have high concentration of calcium ions in this cytosol. Okay, and I'm going to further explain about this muscle contraction, how it happens. Now, in the presence of high concentrations of calcium ion in the cytoplasm, what happens? The calcium ion will bind to the troponin complex. Okay, so on the 18, okay, you can see this 18, they are troponin complex. So this troponin complex has calcium ion binding sites. So when calcium ion binds to this troponin complex via the calcium ion binding sites, this troponin complex will undergo conformational change. Okay, and this will expose the active sites on the actin filaments. Okay, the active sites here refer to the myosin binding sites. Okay, you can see this myosin binding site. Initially, it was being covered by the troponin complex. You can see here. Okay, So binding of calcium ion to this troponin complex will change the conformation of this troponin complex, and this will expose to the myosin binding sites on the actin filaments. Okay, next, what happens after that? Okay, the myosin head, which is in low energy configuration, will bind with an ATP. Okay, you can see here this myosin head will bind to an ATP. Now this ATP, okay, is supplied by mitochondria, okay, via aerobic respiration process. Now ATPase, okay, on the myosin head will split or hydrolyze the ATP into ADP and pi. Okay, when that happens, the myosin head becomes energized and cocked. This is cocked. Okay, initially it was like this in its low energy configuration. Okay, hydrolyze of the ATP cause the myosin head to be energized or has high energy configuration and cocked. Okay, next, the myosin head will bind to the active site or myosin binding site on the actin filament to form a cross bridge, okay, or ectomyosin bridge. The breakdown of ATP, okay, releases pi or inorganic phosphate that triggers the power stroke. So now I'm going to explain the power stroke. Now in the power stroke, what happens? Okay, you can see it's myosin head. Myosin, okay, we pull the actin filament inwards, thus shortening the muscle. Okay, you can see this myosin head pull this actin filaments inwards. Inwards means towards the center of the sarcomere, which is the M membrane. This occurs along the entire length of every myofibril in the muscle cell. Now, okay, after each pull, the myosin head will be in low energy configuration. Now, at the end of the power stroke, okay, we have the myosin head in low energy configuration, right? So the myosin head, okay, detaches from the actin, and the cross bridge is broken when another ATP molecule binds the myosin head. This cycle continues if the concentration of calcium ion is high enough in the cytoplasm. The myosin head will split, hydrolyze the ATP into ADP and inorganic phosphate, become energized and attached to another myosin binding site further along the actin filament and repeat the power stroke. The repeated pulling of the actin by the myosin is known as the Ratchet mechanism. Okay, this process of muscular contraction can last for as long as there is adequate ATP from the mitochondria and calcium ions from the sarcoplasmic reticulum. Once the impulse stops, okay, the calcium ion is pumped back to the sarcoplasmic reticulum and the actin returns to its resting position, causing the muscle to lengthen and relax. Now in that, okay, apparently the muscle cell can no longer produce ATP, right? Because the mitochondria also dies. So the cross bridges cannot be broken, okay, causing the muscle stiffness of death, or also known as the rigor mortis. A living cell always has enough ATP to allow the myosin heads to detach from actin. Okay, now let's put some summary here, okay, on the sliding filament hypothesis. Now, the sliding filament hypothesis describes the mechanism of muscle contraction. During muscle contraction, the actin is repeatedly pulled over the myosin via ratchet mechanism. Okay, and ratchet mechanism is the mechanism where the myosin head from the thick filament attaches to the myosin sites in the actin of the thin filaments and pull it towards the M membrane. Okay, you can see the M membrane here, okay, where this actin is being pulled by the myosin towards the M membrane. Okay, and this is when a muscle contracted. Muscle fibers, okay, shortens and changes in the bending pattern. The thin filaments do not contract or shorten. Please take note on that. Okay, the H zone narrows. Okay, then the I band shortens. The A band, it stays the same. It doesn't change. If you notice here, you compare these two, the A band doesn't change. Okay, the Z lines, however, is being pulled towards each other, right? Okay, so this is a temp how a relaxed sarcomere looks like and a contracted sarcomere. Okay, so that's about it. Okay, see you in the next lesson.